Usually when we talk about stepping into a medical clinic, there is this baseline expectation that the rules of physics and biology are universally agreed upon by the people in charge.
Right, like everyone's operating from the same textbook.
Exactly. You assume a treatment is either a piece of hardware, like a titanium hip replacement, or it's a pharmaceutical drug, like a blood thinner. It feels like the dividing line should be entirely binary.
We desperately want our medical categories to be neat and tidy like that. A device just sits there and provides a structure, and a drug actively alters your biology.
Yeah.
But when you start engineering the actual building blocks of human biology, that clean dividing line just completely shatters. The entire regulatory architecture really struggles to keep up.
And when you dive into the stack of international regulatory frameworks, biotech patents, and medical guidelines we are exploring today, you realize that line isn't just blurry, it is practically a war zone.
Oh, absolutely.
So we're doing a deep dive into this massive stack of source material to answer a deceptively simple question. How on earth do governments decide if a vial of DNA or a syringe full of cells is a simple medical device or a highly regulated biological drug?
It's a huge debate.
It really is.
Yeah.
And to show you just how wild this gets, I want to look at a fascinating global contradiction sitting right at the top of our sources. Right now, in the UK and across Europe, people are walking into aesthetic clinics on their lunch breaks to get their faces injected with salmon DNA.
Yep, the polynucleotides.
Right. They are known as polynucleotides. And legally, across the pond, these injectables are regulated as standard medical devices.
Right.
But if you try to do that in the U.S.,
The FDA is not having it?
the FDA flat out says no.
Yeah.
Those exact same products are not FDA approved for aesthetic injection.
Okay, let's unpack this, because how can the exact same vial of fish DNA be a device in London but an unapproved drug in New York? Well, it forces regulators to define what actually constitutes medicine when the treatment relies on nucleic acids. Like, when you're injecting DNA, regulators have to determine the actual mechanism of action.
Right.
They have to ask, is the DNA acting like a mechanical scaffold in the tissue, or is it acting like a pharmacological agent that's altering the patient's cells?
I want to look closely at the material itself before we get to the legal battle, though. Polynucleotides, often abbreviated as PN, and their closely related cousins, polydeoxyribonucleotides, or PDRN.
A bit of a mouthful.
Totally. But our sources are very clear on what these are and maybe more importantly, what they are not. If you follow aesthetic trends, you might assume this is just, another dermal filler like a cross-linked hyaluronic acid or a neuromodulator.
Right, but these are fundamentally different.
Yeah, we are talking about nucleic acid polymer chains extracted directly from the gonads of salmon or trout.
Which sounds intense, but the manufacturing process detailed in the sources is critical to understanding the regulatory loophole here.
Okay, how so?
Well, the extraction relies on a patented process called PNHPT, which stands for High Purification Technology. The raw fish DNA is subjected to, intense thermal and chemical stress.
So they basically boil it in chemicals.
Pretty much. And the sole purpose of that highly aggressive processing is to denature and strip away every single protein, lipid, and peptide from the source material.
Because if they left that stuff in.
Yeah, if any proteins remain, the human immune system would immediately recognize it as foreign fish tissue and launch a massive inflammatory attack.
Wow.
So by stripping all of that away, the PNHPT process leaves behind a highly purified, biocompatible, ultra-long DNA chain that is essentially immunologically invisible.
Which brings us to the UK and European regulatory stance.
Yeah.
The Medicines and Healthcare Products Regulatory Agency in the UK, the MHRA and the equivalent EU bodies, they look at these purified invisible DNA chains and classify them as medical devices.
Yeah, they just get a CE mark.
Right. Brands like Plinest, Newest, Vitarin, or 70-Hyal are legally marketed with a CE mark or a UKCA mark in Great Britain. They're evaluated for safety, obviously, but evaluated as devices, not drugs.
Which is wild to think about.
I mean, I am looking at this liquid DNA in a syringe, and I have to ask, on behalf of anyone listening, wait, if it's injected into the skin, how is it just a device and not a drug? Is it just because of the intended use?
Well, it comes down to the concept of primary mode of action. The MHRA evaluates how the product actually achieves its intended purpose.
Okay.
So, if a product's action is achieved by physical or mechanical means, it can be a device. These polynucleotide chains are incredibly hydrophilic, meaning they bind massive amounts of water.
Like a sponge.
Exactly. Once injected, they create a three-dimensional polymeric matrix in the dermis. The UK regulators view this as a biological scaffold. Its primary job is to physically hold water and provide a localized structural environment for fibroblasts to migrate into and begin tissue repair.
So it's not actually interacting with the cells directly.
Right. Because the DNA is not entering the patient's cells to alter their genetic code, and it's not binding to receptors to trigger a systemic metabolic cascade, its primary action is deemed mechanical. It's essentially a microscopic injectable sponge.
Wow. Just to note from the sources, those European CE marks for medical devices are currently valid in the UK until at least 2028, after which the UKCA mark takes over entirely.
Good to know.
But I want to pivot to the US FDA, because they look at that exact same microscopic sponge and hit the brakes. There is no polynucleotide injectable FDA approved for aesthetic use.
Right, they have a very different take.
The sources point out that while products like Rezern are cleared by the MFDS in Korea, America just shuts them out of the aesthetic market entirely. If the UK can prove it acts mechanically, why is the FDA so hesitant?
The FDA approaches biological polymers with extreme skepticism. Like, they classify devices based on intended use and indications, putting them into class 1, 2, or 3. But the moment you take nucleic acids derived from an animal source and inject them into a human, the FDA worries about long-term systemic effects.
Even if the manufacturer says it's just a scaffold.
Exactly. Even then, the FDA demands exhaustive proof that these polynucleotides won't degrade into active oligonucleotides that could inadvertently trigger an immune response or display unintended metabolic activity.
So they just don't trust the material.
Basically, yeah. They view the inherent nature of the material as too volatile to simply wave through as a Class 3 device without the rigors of a drug or biologic pathway.
Okay, so the FDA is terrified of purified inert fish DNA just because it's a biological polymer.
Oh, wait. Clinics in the U.S. are injecting people with human stem cells every single day for everything from bad knees to hair loss.
Oh, all the time.
So how does the FDA allow the injection of living, metabolically active human cells if they won't even allow inert fish DNA?
Well, that tension brings us to the FDA's strict fork in the road for human tissues, which is governed by 21 CFR Part 1271.
Right, the HCTP rules. Yeah, human cells, tissues, and cellular and tissue-based products. To handle the absolute explosion of regenerative medicine, the FDA had to create a dual pathway. One path is a low-barrier fast track, and the other is a regulatory mountain.
I am looking at this 21 CFR Part 1271 rulebook, and it outlines the fast track, known as Section 361 of the Public Health Service Act.
The famous Section 361.
Yes. If a clinic's product qualifies solely under Section 361, there is no pre-market safety review. There's no billion-dollar clinical trial.
None of it.
The FDA's only real oversight is ensuring the facility isn't transmitting communicable diseases. It's basically a free pass, which explains why thousands of regenerative clinics claim their products fit under this section.
Right, everyone wants to be under 361.
But to get that pass, a clinic has to satisfy 4 incredibly rigid criteria.
And those criteria are designed to ensure the biological material acts exactly as it did in the donor.
What are the rules?
First, the product can only undergo minimal manipulation. Second, it must be for homologous use, meaning it performs the same basic function in the recipient.
Okay, makes sense.
Third, it cannot be combined with another article, with very narrow exceptions for things like water or crystalloids. And fourth, it cannot have a systemic effect, nor can its primary function depend on the metabolic activity of living cells.
I want to challenge the logic of that third rule, though. Why is the FDA so obsessed with preventing clinics from combining human cells with other substances?
Because combining biologicals with active chemicals or even inert scaffolds can trigger entirely unpredictable metabolic cascades.
Like what?
Well, if you mix a stem cell with a growth factor or a synthetic polymer, you might alter the cell's microenvironment. That stress can cause the cell to differentiate into a different tissue type or hyperproliferate.
Oh, wow. So it changes how the cell behaves.
Exactly. The FDA's stance is that the moment you introduce a foreign element, you can no longer guarantee the cell will behave as it naturally would, which completely voids the safety profile of a standard tissue transfer.
So if we look at the first rule, minimal manipulation, Section 361 is essentially like taking a piece of wood from a tree and using it to build a cabin. you haven't fundamentally changed the wood.
I love that analogy. Yes, you haven't changed it at all.
Right. But what happens if you take that wood, grind it down into sawdust, mix it with the industrial adhesives, press it under extreme heat, and make particle board?
The wood analogy perfectly illustrates the biological reality here. If you turn that wood into particle board, you have fundamentally altered its original relevant characteristics.
It's not just wood anymore.
Right. The FDA categorizes that as more than minimal manipulation. And the moment you cross that line with human cells, you are violently ejected from the Section 361 pathway and pushed into Section 351. Meaning you are no longer a tissue, you are a biological drug.
And the sources detail exactly how clinics trip over this line in practice. Let's say a clinic takes your autologous stem cells, but instead of just re-injecting them into your knee, they put them in a Petri dish and culture expand them to grow a larger dose.
Which happens a lot.
Right. But the FDA says that in vitro expansion is more than minimal manipulation. Why? What is actually happening to the cells in that dish that terrifies regulators?
Well, extended culturing places immense selective pressure on human cells. In a Petri dish, devoid of the normal physiological cues of the human body, cells are forced to adapt to a synthetic environment.
And that adaptation is dangerous.
Very. Our sources show that this adaptation leads to genomic instability. The telomeres might shorten prematurely. The cells frequently lose critical surface markers that dictate how they communicate with other tissues.
So they're basically going rogue.
In some cases, yes. The aggressive proliferation required to expand the culture can cause mutations that actually mimic oncogenesis or tumor formation.
Wow.
Because of those severe risks, the FDA demands that any culture-expanded cell go through the investigational new drug application process, culminating in a biologics license application, or BLA.
Which costs millions of dollars and takes a decade.
Exactly.
This brings up a critical observation from the sources regarding how these cell therapies are marketed directly to patients. You see clinics claiming their expanded stem cells are vaccine-free or harvested pre-COVID or marketed simply as completely natural.
Oh, the marketing is wild.
They plaster these terms on their websites to imply the product is inherently safe and shouldn't be scrutinized by the FDA.
It is a deliberate attempt to bypass regulatory reality using branding. I mean, the law does not care if the original source was natural.
Right, it's about what you did to it.
Exactly. Classification is dictated entirely by the scientific manipulation the material underwent. If a clinic culture expands a completely natural autologous cell, the resulting genomic instability makes it a Section 351 biologic. Marketing copy cannot overwrite the cellular changes that occurred in the lab.
So the U.S. is locked in this intense, rigid battle over the 361 versus 351 divide. If you manipulate a cell too much, it's a biologic drug. But if the U.S. lumps everything manipulated into the biologics bucket, how does Europe handle the absolute chaos of living cellular treatments?
They took a completely different approach. They recognized that forcing engineered human tissue into the traditional pharmaceutical drug paradigm was fundamentally flawed.
Because it's alive.
Right. A living cell does not behave like a static chemical compound. So to solve this, the EU constructed a bespoke regulatory architecture called Advanced Therapy Medicinal Products, or ATMPs, governed by EU Regulation 1394/2007.
And these are evaluated differently.
Yeah, instead of standard drug evaluation, these are evaluated centrally by the EMA's Committee for Advanced Therapies, the CNET.
And the brilliance of the EMA is that they broke these ATMPs down based on what the cellular material is actually doing.
Yeah.
They created 4 distinct subcategories, which is so smart.
It really is.
If you are administering genetic material to alter a biological sequence, you fall under Gene Therapy Medicinal Products, or GTMPs. If you are manipulating cells to treat a disease, you are looking at Somatic Cell Therapy Medicinal Products, or SCTMPs.
Right.
But the third category is where the EU really differentiates itself from the U.S. Tissue Engineered Products, or DAQs.
Yeah, the lack of a dedicated tissue-engineered classification in the U.S. causes immense friction for developers.
How so?
Well, by defining a TEP as engineered cells or tissues specifically used to regenerate, repair, or replace human tissue, the EU carves out a protective space for regenerative medicine.
Oh, I see.
They acknowledge that growing a patch of living cartilage in a lab to replace a damaged meniscus operates under different scientific principles than, say, injecting a viral vector to alter someone's genome.
That makes total sense.
Furthermore, they created a fourth category, combined ATMPs, or CATMPs.
Right. The combined ATMPs are exactly what they sound like. It's where you take one of those cell therapies and combine it with a medical device, like a synthetic polymer scaffold, as an integral part of the product.
Yep, bring the two worlds together.
But even with these highly specific buckets, biology is messy. Like, if a biotech startup invents a borderline product, say, a cell that alters genes, but also acts as a structural tissue replacement, how do they know which category applies before they spend millions on trials?
That's the million-dollar question. Because the science inherently outpaces the legislation, the EMA instituted a critical lifeline known as the Article 17 procedure.
How does that work?
An applicant developing a borderline product can submit their data and request a binding scientific recommendation directly from the agency.
And they actually give an answer?
Yes. The EMA is legally required to consult with the Commission and deliver a verdict within exactly 60 days on whether the product is an ATMP and precisely which subcategory it falls into.
That 60-day rule is a game changer. I mean, when developers aren't guessing about their regulatory obligations, they are significantly more willing to secure funding and run the trials.
Absolutely.
The sources highlight the impact of this framework. There are currently 23 cell and gene therapy products approved for use in the UK alone, with dozens more moving through clinical development. That level of regulatory clarity directly dictates whether a life-saving treatment reaches a patient or dies in a boardroom.
If we connect this to the bigger picture, it proves that biological regulation cannot be reactionary. The EU framework succeeds because it anticipates the complexity of living medicines rather than trying to force them into historical drug definitions.
Okay, we've covered the mechanical action of polynucleotide scaffolds hydrating the skin, and we've navigated the minimal manipulation rules for human cells. Let's bring this all together.
Let's do it.
What happens when developers move past simple mechanical scaffolds and engineered tissues and purposefully design nucleic acids to weaponize the body's immune system?
That is the exact moment the regulatory boundary between a structural device and a systemic biological drug completely evaporates.
Here's where it gets really interesting. Digging through the source stack, we have an actual U.S. patent.
Yeah.
Patent number 8,283,328B2, filed by Arthur M. Krieg and colleagues at Coley Pharmaceutical Group. The title tells you immediately we have left the realm of aesthetic skin hydration.
What's the title?
Immunostimulatory nucleic acids. The patent details the creation of soft or semi-soft CpG immunostimulatory oligonucleotides. We are talking about short, highly synthetic DNA or RNA molecules.
And when you look at the raw data charts provided in this patent, the biological mechanics are staggering.
Tell me about it.
These synthetic oligonucleotides are designed with unmethylated CpG motifs. In nature, these specific DNA sequences are abundant in bacterial and viral genomes, but very rare in human DNA.
So they look like an infection to the body.
Exactly. By synthesizing these sequences, the developers are actively tricking the human immune system's toll-like receptors into believing the body is under a massive microbial attack.
The graphs in the source confirm exactly that. These synthetic molecules aren't just sitting passively in the tissue. They trigger huge dose-dependent spikes in specific cytokines, namely interferon alpha and interleukin-10.
The biological significance of those spikes cannot be overstated.
What do they do?
Well, interferon alpha is a primary driver of the antiviral immune response, commanding cells to halt protein synthesis to stop viral replication. IL-10 is a master regulator that prevents the immune system from causing catastrophic inflammatory damage to host tissue.
So it's like the gas and the brakes at the same time.
Exactly. By manipulating these cytokines, the synthetic DNA is actively dictating the immune environment.
But the patent data goes even deeper than cytokines. The charts show these nucleic acids binding to and activating CD69 on natural killer cells, alongside massive spikes in CD80 and CD86 markers on antigen-presenting cells.
CD80 and CD86 are critical co-stimulatory molecules. Spiking those markers isn't just sounding a general alarm.
No.
It's like the synthetic DNA is actively handing out the launch keys to the T cells, providing the exact molecular signals required for them to deploy their targeted weapons against an assumed threat.
This proves exactly why the FDA distinguishes products based on intended use and mechanism of action. I mean, when a synthetic polynucleotide is engineered to bind to toll-like receptors and hand launch keys to T cells, it violates every single rule of a medical device or a Section 361 tissue.
Oh, 100%.
Its primary function is undeniably pharmacological and immunological. It relies entirely on the metabolic activity of the patient's living immune cells to achieve its purpose.
It leaves regulators with zero ambiguity. A product exhibiting this level of systemic metabolic interference crosses a hard boundary into a biological drug.
It has to.
It demands the deepest level of scrutiny a new drug application or a product license application because the potential for runaway autoimmune cascades or cytokine storms is just immense.
Which makes total sense for isolated synthetic DNA. But this raises an important question about the immediate future of biotech. Scientists are no longer working in isolation. They aren't just making inert structural scaffolds, and they aren't just making immune-triggering drugs. They are combining them. They're weaving highly active biological components directly into simple device-like structures.
And that is the ultimate nightmare scenario for a regulator.
I can imagine.
Just imagine a 3D printed synthetic mesh, which is technically a device. Now coat that mesh in genetically modified human cells, which is an ATMP or a biologic.
Okay, this is getting complicated.
It gets worse. Finally, program those cells to slowly release immunostimulatory oligonucleotides to prevent rejection, which acts as a pharmacological drug. What exactly is that product?
Oh, man.
The U.S. intercenter jurisdictions would fight over whether it is a device or a biologic. The EU might streamline it as a combined ATMP, but the friction between the explosive speed of biological innovation and the inherent rigidity of medical law is going to cause massive regulatory bottlenecks.
It really pulls back the curtain on the invisible war happening behind every medical breakthrough. We started with the paradox of salmon DNA acting as a microscopic sponge under a UKCA device mark.
A long way from where we ended up.
Definitely. We navigated the FDA's strict rules on how a petri dish turns a human cell into a heavily regulated drug, explored the EU's proactive ATMP categories, and finally broke down patented oligonucleotides designed to systematically hack our immune responses.
That's a lot to take in.
It is. So, what does this all mean for you the next time you are scrolling through the news? It means when you see a headline promoting a miracle stem cell cure at a local strip mall, or a clinic advertising a natural DNA skin booster, you are no longer just seeing the marketing.
You know what's actually going on.
You understand the intense scientific and legal battles determining whether the syringe in that picture holds a simple structural device, a minimally manipulated tissue, or a highly active systemic biological drug masquerading as a quick fix.
It also leaves us with a profound reality to consider regarding the trajectory of modern medicine.
What's that?
As technologies like 3D bioprinting become commercially viable, we are rapidly approaching an era where we will be printing functional organs using a patient's own genetically modified cells.
Wow, yeah.
When a bioprinter produces a living, beating heart in a lab, the regulatory frameworks of today will completely fracture. Is the printer a medical device? Is the bio-ink considered a Section 351 biological drug? And is the final printed organ an advanced therapy medicinal product, or is it simply a human organ?
That is mind-bending.
The regulatory lines defining what constitutes a medicine won't just be blurred in the future. They will likely have to be erased and rewritten entirely.